Thank you all for joining us for another amazing session of BrainX Community Live for the month of June in 2022. For those of you who are joining us for the first time, we started BrainX Community 3+ years ago with the idea of having a platform for machine learning in healthcare for good. And over the time period, we have grown to 4,000+ member international community and growing strong. There is a very large and active LinkedIn group with the same name of BrainX Community. So if you are on social media, please join us over there. We share a lot of information and knowledge, and I'll talk more about it. Our website, brainxai.org, so you can join us through our website too. And then we do the live monthly sessions over here, just like we're doing today. This is our website where you can land on the homepage, and it has all the key updates, so you can stay updated about various resources that we have. We have the Connect section, that is where we have our monthly sessions and prior sessions information. And don't worry if you miss any of the sessions, you can go to our YouTube channel where all these are posted. So you can watch all of our YouTube recorded sessions even later. Please subscribe the channel so that you get information about the latest posting over there. As I mentioned, a very active LinkedIn group. We share a lot of articles over there, information related to meetings, information related to opportunities. So please join us there too. And then we have the Learn section on our web page where we provide curated and categorized information about links to some of the very interesting articles. And both the articles that are going to be discussed today are actually posted over there. So we provide direct links to those articles that have been cited heavily or have gained a lot of interest. And you can go to the websites directly and read those articles from here. And of course, a lot of journals are now focusing on AI in healthcare. So if you wanted to know which journals you should follow, you could go to our Learn page and you could filter it by journals, and you'll get the selection of key journals that are focused on AI in healthcare. We created a new book section, so there are a lot of books getting published in this, and if you want to keep track of that, you can filter our Learn section for books and you'll find those there. And then everybody's looking for data. So if you're looking for data, which is open source, whether it's imaging or cardiology or critical care, you can go to our data section. It is the largest repository of links to open source data sets. Of course, we cannot provide you data sets directly. So we provide you with curated, annotated links, whichever specialty you're looking for, and you'll find that you can go to those data sets directly from this resource. And then something very exciting that we started last year was to showcase some of the eminent people who are working in AI in healthcare. We started our podcast series called BrainX Talks, thanks to Ashish and Alok who have joined us. And you can listen to the views, the visions, the experiences of some of the eminent scientists, both physicians and engineers and data scientists through this podcast series. So please subscribe our podcast called BrainX Talks and get to listen to their experiences and visions. Of course, now a lot of meetings are back and are happening in person, and I'm sure you want to reconnect. So if you want to know where you could reconnect next, so go to our website and look at the meeting section or conferences section, and you'll find a whole list of meetings focused on AI in healthcare. And thereby we can connect and network at some of these meetings and foster opportunities to collaborate in the future. And earlier in the year, we released our 2021 year in review, which was very exciting. And based on the theme this particular month, critical care, although has large amount of data sets which are open source, is still struggling to produce a lot of research work and publications in this area. But again, the two speakers who are going to focus on the AI in critical care have shown that amazing amount of research can be done and the applications built to help our critically ill patients. So with that, I'm going to introduce our speakers. Our first set of speakers is Dr. Robert Stevens. He's the Director, Division of Informatics, Integration, Innovation. He's also the Director of Precision Medicine. He's the Associate Professor at Johns Hopkins University. He's a close friend. He's also, along with me, the chair of the Data Science Knowledge Education Group for the Society of Critical Care Medicine, very focused on AI applications in healthcare. So we'd love to hear his thoughts. And then we have him being joined by Shubhayu Bhattacharyay. He's the BS Gates Scholar, PhD candidate at Cambridge University, who's worked with Dr. Stevens and the team and their publication related to the time series for critically ill patients. Followed by their presentation is Baptiste Vasey, who is going to join us. He's also a DPhil candidate from Nuffield Department of Surgical Sciences, University of Oxford, and a Berrow Foundation Lord Florey Scholar at Lincoln College. He's joining us from across the pond, and we are very excited to have him here as he led the very important work on Decide AI, which is for a framework for evaluating AI applications, just like Robert Stevens and Shubhayu have been working on. So we will learn how to evaluate some of these applications through him. So with that introduction, I'm going to turn over to Dr. Stevens.
Thank you so much, Dr. Mather, for that introduction. I'm going to share my screen. Can you see my screen now? Yes?
Yes.
Okay. So I would like to thank you for the invitation, as well as for your fantastic efforts creating this BrainX community. It's a really remarkable achievement and a beautiful example of what can be done, in terms of connecting researchers and clinicians across the world around this theme of leveraging data science to advance health. So I'm going to talk today about this work that was conducted a couple of years ago at Johns Hopkins University, where I currently serve on the faculty as well as a attending intensivist and anesthesiologist. I'd like to recognize Shubhayu, who's going to be actually presenting most of the results of this work. Shubhayu actually graduated from Johns Hopkins University 2 years ago with a degree in biomedical engineering, as well as in applied math and science. And he conducted this work with my group over the 2 last years of his undergraduate career. He's now a Gates Scholar at Cambridge University in the Department of Anesthesia and Neuroscience, and is conducting work over there that is also of great importance. So this is the paper that we're going to be referencing today. It was published a few months ago in Nature Scientific Reports, and I'm providing you the link here. The key people on this paper are Dr. Ralph Etienne Cummings, who is professor of electrical and computer engineering here at Johns Hopkins University, and really a leader in terms of developing very, very large systems integration to better measure and modulate neural function in humans. Dr. Etienne Cummings is a close friend and colleague, and he co-mentors a number of students, including the photo that you can see on the bottom left-hand corner of this slide, which is John Rattray. John Rattray is currently a PhD candidate in electrical engineering here at Johns Hopkins University, and is also one of the key contributors to this paper, along with Shubhayu, who I already introduced to you. So when we think about severe brain injury, this is a big problem. The World Health Organization estimates that if you take stroke and traumatic brain injury together, they are the leading causes of disability across the world, and among the leading causes of death. According to the most recent statistics, around 17 to 18 million people have a stroke each year, and this leads to upwards of 6 million deaths. So it's the second leading cause of death across the world, and it's the leading cause of disability. Traumatic brain injury, less frequent, but also a very, very important cause of death and disability, especially among younger individuals, children, and young adults. And then cardiac arrest, which is also one of the leading causes of long-term neurological disability globally. So although these are major problems, the issue is that we are not doing a great job of taking care of patients who suffer from these injuries, where in spite of many advances, our ability to accurately detect and diagnose brain injury is constrained. Our classification systems are not anchored in biological systems. We don't have effective therapies. We have very good supportive intensive care, but we don't have effective therapies to really repair the brain. We're not very good at predicting and prognosticating. We are not good at all at preventing neurological injuries and preventing neurological worsening. And we are not terribly good either at achieving meaningful long-term recoveries in many, many cases. So I'm just going to give you some examples. And this is a paper that was published by a colleague of mine, some colleagues of mine at Columbia University, simply demonstrating that if you take patients who have a severe brain injury are in a coma in the intensive care unit, many of these individuals, about 15 to 20% of them, actually have significant cognitive processing abilities. So if you use EEG and you apply machine learning, in this case it was a random forest classifier, you will find that a significant subset, close to 20% of patients, actually retain conscious processing in spite of being clinically unresponsive. So this is an example of misclassification. If we consider the this next slide, this here we're showing you essentially different prognostic scores, right? So across different types of brain injury, stroke, cerebral hemorrhage, subarachnoid hemorrhage due to aneurysmal rupture, traumatic brain injury, post-cardiac arrest encephalopathies. On the right hand of this, this table, you can see the predictive values. So many of these are expressed as area under the receiver operating characteristic curves. And you recall that this goes from 0.5 to 1, 1 being a perfect classifier and 0.5 being a classifier that has no value. You can see that most of these, the current state of the art in terms of prognostication, we're achieving around 70 to 80%, 0.7, 0.8 in terms of area under the receiver operating characteristic curve. So it means that 20 to 30% of the time, the best that we have in terms of prognosticating is unable to accurately classify patients with severe brain injury. So if we consider, therapies and clinical trials, this is a summary of many, many different studies conducted in patients with traumatic brain injury, cerebral hemorrhage, aneurysmal subarachnoid hemorrhage. And you can see, if you look at the right-hand column again, the treatment effects, for these different interventions, these different treatments that have been tested in randomized controlled trials are largely inconclusive or even harmful, right? So this shows us that essentially we're not terribly good. And this is a slide illustrating this concept of a translational gap, right? So many treatments that have been tested in preclinical models in animals have been shown to be highly effective. So the response ratio is expressed here on the left-hand side of this slide. Essentially, the lower the response ratio, the better the treatment is in experimental studies. And you can see that as we move from preclinical model systems, so animal studies essentially, towards clinical trials, the efficacy is progressively lost. If you look at the right-hand part of this slide, you can see that in phase 3 clinical trials, these interventions are largely ineffective. So there's a huge gap between the effectiveness of interventions in preclinical systems and their effectiveness in clinical trials. So the question is really, what did we get wrong and where do we go from here? So I'm going to make a little bit of a leap of faith here and propose something that I feel very strongly about and that I've been heavily invested in for the past several years, which is precision medicine. The provocative hypothesis of precision medicine, as many of you know, is that if we combine, clinical intuitions, knowledge of medicine and biological hypotheses together with access to high resolution, high dimensional data sets, together with processing capability largely due to machine learning, we can potentially begin to solve some of these important problems. And really the precision medicine paradigm is in many ways a cycle, right? So you start off with those things that I mentioned, domain expertise, data, artificial intelligence. This helps you to, through a process of iterative computation, to arrive at some insights. Using those insights, you can then better classify and predict. You can design interventions, and you can hopefully achieve better outcomes. And then what's kind of virtuous in this cycle is that the data collected with those outcomes can then be fed back into the cycle to advance the science. So when we talk about high resolution data or high dimensional data, we are, many people think of precision medicine as essentially genomic medicine, but it's a lot more than just genomics, right? It includes things like the epigenome, the changes in protein expression, in metabolic expression, things like the microbiome. My group has been particularly interested in physiological time series, so we can call it physiome, as well as the phenotype, the clinical phenotype, which is in some ways we can refer to as the phenome. And we're going to be focusing in this session on one of those phenotypic expressions, which is motor time series. So really the sort of fundamental insight for this work is this notion of motor outputs or motor phenotypes, right? So the hypothesis that we posed from the beginning was that the motor expression of critically ill brain injured patients is a sort of window on their underlying neurological state, and maybe even also on their underlying physiological state, right? So that we can gain fundamental insights on what's happening in the brain, what's happening physiologically in our patients by analyzing the motor activity, the motion time series of our patients. And we can use this with artificial intelligence. We can use this for the purposes of classification and prediction. So really the sequence that we went through, and Shubhayu is going to go through this in a lot more detail, is indeed this quantification of motor phenotypes, the generation of these time series, engineering of features, and then the use of computational analysis. So I'm going to hand this over now to Shubhayu, who's joining us from Cambridge, where it's quite late. I think it's past 11 o'clock. So thank you very much, Shubhayu. And I'm going to come out of this presentation so you can share your slides.
Excellent. Thank you, Dr. Stevens. Let me share my screen. 1 second. Okay.
If you like, you want me to share them, your slides, or?
I think I can do it. If I can. Actually, yes, if you're able to do it, Dr. Stevens, that'd be more convenient.
Okay. Let me get to it. Yeah,
thank you.
So.
Yeah, I think I didn't realize that Shubhayu is across the pond.
Yep, here we go.
Like
so this is your
thank you for joining us.
Oh, my pleasure. Thank you, and good afternoon and evening to everyone. So building off of what Dr. Stevens mentioned, I'll go into a little bit more detail into our study and how we tried to break down and achieve some of those big picture aims. So we started in the Johns Hopkins Neuro ICU. We had a relatively general recruitment criteria. We enrolled adults of at least 18 years who presented with an acute injury or illness to the brain that resulted in impaired consciousness. And we also selected for patients that had both upper and lower limbs and had no apparent lesions or injuries in these areas in which we would place wearable sensors. And we also selected patients who were not expected to die within or have withdrawal of life-sustaining sustaining therapies within 24 hours of enrollment. And so, let's see, and then the next part. So this resulted in a study sample of 69 patients who we followed through their ICU stay in the neuro ICU up to a year. And as you can see in this diagram, we placed 6 wearable sensors bilaterally on the elbows, wrists, and ankles of our study sample. And we also placed an additional 7th sensor at the foot of the bed for external movement correction, which I'll get into in a bit. But as you can see, we collected 3 axes of accelerometry from each of these 7 sensors, and we collected or we sampled at 10 hertz. We ended up collecting about a day's worth of information from each patient. So the next part shows that the median continuous recording duration was about 24 hours. And so this meant at 10 hertz, we collected a lot of accelerometry for each patient during different moments in the neuro ICU. So then the question was, how do we break down these waveforms into something that could be clinically useful or interpretable? So first, we processed the accelerometry. Using a high-pass filter, we removed low-frequency noise, and you can see that this removes the offset effect of accelerometry and allows us to focus on those dynamic events that correspond to intentional movement in accelerometry. And then in 5-second non-overlapping windows, we extracted a bevy of motion features. And as this next part shows, we extracted 6 motion features that correspond to different domains. So if Dr. Stevens, if you would click one more time. Yes, so this table represents the different motion features we collected or we extracted from the accelerometry. And our motivation to extract these features is that we wanted a diverse representation of signal processing domains. So you can see that we have the time, frequency, and wavelet domains. But we also wanted features that were high performing in previous activity classification studies. So part of our hypothesis was extending activity classification to the clinical setting, not for classifying direct motion activities, but corresponding these features with neurologically significant outcomes and markers. And so in the next part, we examine the feature set here in this data frame. And we see that, especially in that middle column, that there are significant challenges of collecting this sort of high resolution information in the intensive care unit. As with any other modality, there are going to be missing values. And in our case, it was not uncommon due to patients having to be taken in and out of the room or other interruptions for clinical reasons. So for our case, we imputed with a stochastic multiple imputation time series algorithm called Amelia-2 and produced 9 independent stochastic imputations of our data set. And then we corrected for the bed motion. And the way we did this is we identified moments in which the bed motion exceeded a threshold of signal magnitude area represented by SMA. And this is according to the literature, of course, this threshold corresponds to the threshold between static and dynamic activity in accelerometry. And so if we identified points in which the bed exhibited dynamic activity, we would then investigate and see if patient motion preceded that point or not. And if patient motion didn't precede that point, we would subtract the contribution of bed activity from each of the limb features, from each of the body sensors. So what we're left with for each patient is essentially 42 time series, actually 42 times 9 for each of those imputations. So then the challenge remains, how do we further reduce this data into something that could be useful or interpretable? And so what we did here, we represent an entire day's worth of accelerometry starting from 0 to 24. And then what we did is we automatically extracted from the electronic health records each time that a Glasgow Coma Scale or neurological assessment was taken on the patient. And those are represented by these red arrows. And then what we did from there is we extracted an observation window's worth of accelerometry corresponding to or directly preceding each of these Glasgow Coma Scale assessments. Now, the observation window itself was a variable in our study, and we evaluated 18 different observation windows ranging from 3 minutes to 24 hours directly preceding each of these assessments. And here in an example of one of the observation windows, we show what we did with that information. With those 42 time series, we in fact started by adding another feature into the mix, a simple feature called proportion of dynamic activity, which just measures it's a summary statistic for the entire observation window for how much dynamic activity there was. And then we performed a supervised dimensionality reduction algorithm called LOL, or linear optimal low rank projection. And this enables us to take those 42 time series and reduce it to a single low dimensional vector for each patient. And from that low dimensional vector, we, in the next part, we evaluate the prognostic power or the detection power of this vector through logistic regression for 3 different model endpoints, for 3 different tasks. The first task was whether or not this motion, this physical activity motion or information collected from accelerometry could detect the concurrent motor component of the Glasgow Coma Scale, which is our proxy for the neurological state of motion. As well as predicting short-term and long-term functional outcome according to the Glasgow Outcome Scale. And I'll talk about each of those in a bit, but it's important to note that we perform this in a cross-validation environment on our prospective cohort. So in the first task, we try to detect the best motor response, so the neurological function at that time. And as you can see in the bottom right table, the Glasgow Coma Scale, or the motor component of the Glasgow Coma Scale, is a 6-point ordinal scale, which maps the patient's best motor response or neurological motor function based on their response to a stimulus, a painful stimulus during a neurological exam. And so what we observe at each of the thresholds of GCSM is that our model achieves a significant signal, well, except for GCSM greater than 2, for the remaining thresholds, our model achieves a significant signal of AUC 95% confidence greater than 0.5. But if we look at the trajectories in the short-term observation windows, we see in the lower left that only for GCSM greater than 4 does the model achieve some sort of regular or consistent pattern of significant detection power over the shorter observation windows. And this is significant because GCSM greater than 4 corresponds to a patient's neurological ability for intentional movement. And so this identifies that those physical activity markers that we extracted from accelerometry are able to detect this clinically important threshold of neurological state. And then in the next task of predicting functional outcome at ICU and hospital discharge, we see that the model fails to achieve any sort of significant power except for that GOSC greater than 5 threshold. We don't go above the GOSC greater than 5 threshold because we didn't have enough patients with good recovery at hospital discharge, as we started with a very severe cohort to begin with, severe brain injury cohort. But we observed this rather strong prediction power for GOSC greater than 5. And we see in the bottom left, in terms of that this is achieved with a 2-hour observation window. And so we observed that remarkably with longer observation windows, of course, predicting functional outcome is a more challenging clinical task. But with a longer observation window of accelerometry, there is some sort of significance in the signal between these motion features and the functional outcome. And then if we specifically look at the motion features, we then investigate the relative feature importance in the different tasks. So this is a matrix that shows, on the left-hand side, we have the 8 different features, and on the bottom, we have the 6 different sensor placements. And in the combination of these, we calculate a significant score based on that reduction or that LOL supervised reduction algorithm. And we see consistently that there's more variance across the feature types than there is within or across the sensor placements. And this is important because it argues for a multidimensional look at motion for clinical assessment, not just looking at time domain or frequency domain or wavelet domain features, but looking at a combination of these features and understanding their specific value added for each sensor placement. And if we just look at these most important features based on this matrix, the top one, PDA, as I said before, is a summary statistic that's often used in wearables and actigraphy to summarize a crude measurement of overall activity. And this, of course, it was important for GCSM detection, as GCSM greater than 4 represents that ability for intentional physical activity. But then if we look at frequency domain entropy in the frequency domain, FDE, this had a relatively high impact as well. And FDE is used in accelerometry to discriminate simple acceleration patterns from more complex patterns. And this adds, in this way, the model is able to encode some of the complexity of motion patterns, not just the overall level of physical activity, but the frequency and the complexity of that frequency in detection. And we see a similar story in GOSE prediction as well, where there's more variation across the feature types than there is across the sensor placements. And a consistent outcome across the two prediction tasks is that in the high and low frequency pairs, HLF-H and HLF-L, we see more significant contribution from the high frequency component of accelerometry features. And this motivates us to search into those finer movements, such as those such as tremors or wrist movements that help distinguish or discriminate patients of different neurological states and eventual functional outcomes. And so this allows us to decode some of the patterns of movement that correspond to functional states, as well as outcome in brain injury. And then in this slide, we show one of the individual cases, one patient during our recording period experienced a shift between GOSE3 and GOSE5. GCSM3 and GCSM5. Of course, this is across the threshold of motor function in which our model performs best. And as you can see, that the observation window has a direct effect on the sensitivity of the model to changes in GCSM. The shorter observation window of 27 minutes is much more sensitive, but it's more sporadic, whereas the longer observation window of 6 hours has a sustained increase. Now, this patient, beyond this window that we show, experienced an improvement, a general improvement, and a sustained level of GCSM5 and then eventually 6 before ICU discharge. And it shows that our accelerometry-based features were able to detect this upswing in motor function before, a few hours before that clinical assessment. And so this is an argument for continuous monitoring of motion and the clinical information that we can yield from this modality. Of course, we only test accelerometry in isolation. We only test accelerometry, or we only put accelerometry on trial. But it is promising to see that there is a signal based off of which we can integrate this modality into larger dynamic models and incorporate a continuous assessment of motion into our bigger picture of severe brain injury care.
So thank you, Shubhayu. I just want to conclude with one or two slides to just summarize this work. I think, this is certainly just the beginning of a story that we think is ongoing, but our feeling is that these quantitative motor features are biomarkers. They're biomarkers of neurological function, as evidenced by our ability to sort of predict and map onto these transitions in the state of level of consciousness, as well as predictors of neurological outcomes, at least in the short term. And we're building on this work now to integrate motion into multimodal assessments and monitoring paradigms. And so here I'm showing you on this final slide a sensor that is being designed by John Rattray, the PhD student here at Hopkins that I referred to earlier, that includes, in addition to accelerometry, which is the primary modality for the published study, EMG, right? So we're able to record muscle activity, as well as photoplethysmography, so PPG, electrodermal activity, and also electrocardiography. So this multimodal sensor, which is currently being tested and we're hoping to deploy in a clinical study at Johns Hopkins in the next couple of months, hopefully will allow us to derive multiple simultaneous time series physiology with the goal of even further enhancing our ability to classify and predict. So with that, I'd like to conclude with acknowledgement of collaborators here at Johns Hopkins, as well as in many other institutions. Funding for this work, which was in part from Johns Hopkins University. And then this is the final slide with links that are available in case you're interested to learn more about this work and connect with me or with Shubhayu. So with that, I will stop, and I believe the questions will come at the end. Is that correct, Piyush?
Yes, Robert. I think we'll take questions at the end, but this was just amazing. I learned so much. So thank you, Shubhayu and Robert, for this awesome presentation. A lot of thoughts come to my mind, but let's have Baptiste join us and tell us about the Decide AI and how he thinks or he conceives amazing work like this can be evaluated using the framework that has been proposed and published recently. So Baptiste, I'm going to turn it over to you for the next presentation on Decide AI.
Thank you very much. Let me just share my screen.
Yeah, a lot of questions, very impressive and interesting work.
Okay, I believe you can see this, the full screen now?
Yes, that's correct.
Perfect. So thank you very much, Dr. Matter, for the invitation to speak tonight and present the Decide AI reporting guideline, which had the opportunity and the pleasure to develop with yourself and many other collaborators across different expertise related to AI and AI development. So Decide AI is a new reporting guideline, which is one of the first stage-specific guidelines. I'll come back to that later in the presentation, focusing on the early stage live clinical evaluation of AI systems. And quite interestingly, I will start my presentation with the same observation as Dr. Stevens for AI, that we have now a growing number of evidence that those systems, so AI systems, are efficient in preclinical studies. And here I took just a few examples. There are many, many more. We have also systematic reviews and meta-analysis showing equal performance between deep learning models and human physician. But as we progress through the evaluation pathway, as we progress through actual clinical settings, then those evidence become rarer and rarer. And I could find during all my PhD only very few evidence of clinical efficacy as evaluated through robust randomized control trial. Again, that's a slide from another presentation. I won't go through all of them, but basically I could count on my two hands the number of solid large-scale randomized trial using an evaluating AI system in actual clinical settings. More and more are coming, but there is still a huge discrepancy between the literally dozens of thousands of papers. I think in 2000 last year, the mesh term artificial intelligence in Medline returned something like 21,000, 23,000 papers, and we have only a handful of those robust trials at the end of the day. And so the question is why do we see this gap? Why do we see this discrepancy? And that's where my main supervisor, Professor McCulloch, at the University of Oxford, and myself started thinking, okay, how exactly are those technology evaluated from the very first development, the very first ideation to the large-scale trials that we consider as gold standard in medicine. And while thinking about it, we try to compare the development of AI system to the development of other medical innovation, which are better known and more standardized so far. And the first one is, of course, drugs, which has a very standardized, very well-known development pathway. We start with preclinical development, animal studies, moving on to phase 1, phase 2 trials, which are small-scale efficacy, utility, and safety trial, to the phase 3 trial, which are the large-scale effectiveness trial, and the post-market surveillance coming in phase 4. I think maybe more interestingly for the development of AI system was the parallel to surgical innovation, and that's was the original brainchild of my supervisor, the ideal framework, which was a framework developed to guide the evaluation of surgical innovation of new procedures, taking into account that unlike drugs, if you want to evaluate the effectiveness of the new procedures, you need to take into account not only the variability in the patients you apply the procedure to, but the variability in the surgeons that are delivering the procedures. The procedures will therefore not be a fixed intervention as would a drug be, but would vary on the people that are actually delivering it. And you can make a parallel to AI in a sense that as we for the first time have an intervention or a system which represents a competing or collaborating form of intelligence, the way clinicians will respond to this collaborative or competing form of intelligence will depend on their own characteristic, their level of experience, their personality, their previous exposure to digital technology. So we made those two comparisons and realized that there was a big gap in the middle of what existed currently as guidance to develop AI system and evaluate them and report them. At the very beginning of this development pathway, you have the in silico evaluation and the silent and shadow mode evaluation. And for that, at the moment, two major reporting guideline extensions are being developed, which are Tripod AI and Start AI, which you might have heard of. And on the other side, on the side of large-scale randomized trial, and that was the first big AI-specific reporting guideline to be published in Nature Medicine, Lancet Digital Health, and the BMJ last year. That's the Spirit AI and Consort AI guidelines. But in the middle, there was this big gap, which was filled by other development pathways, like drugs and surgical innovation, but not for AI in healthcare. And so we decided to, well, dig a bit deeper and try to see if that would be interesting to develop some guidance for this specific phase of the development pathway, and to think about why exactly is this phase important. And the answer we had to this question is it's important for four main, in our view, four main aspects. The first one is to analyze the performance of the system with human users. Because as I said, if you have different form of intelligence that might compete, you might get you might get a gap in performance between what the system, what your AI system is outputting, and what the final decision is. In the sense that if you have a human clinician in the middle, and especially a human clinician that will remain liable, at least in the near future, for the decision making in clinic, this human clinician will sometimes override the decision of the AI system, sometimes trust it, sometimes not, but might influence it and modify it. So I think it's unreasonable to think that the performance you see in silico studies for defined AI system will be the one it will have in clinical setting. So we need to evaluate that, and we need to evaluate that at small scales first, in order to, one, not waste resources testing systems that are not actually efficient when used with human, and to avoid to expose patients to unknown risk before having been tested at the small scales, as I said. which link to the second point. We need to ensure patient safety. We need to ensure that those models are sure in the decision they make and leads human clinician to make. And we need to do that at small scales. A third major and important point is the evaluation of human factors. Human factors is the field that looks at all the external factors that will influence human either cognitive or physical performance. Human factor research is well developed and well known in other research area and other sector of industry. All the safety critical domains like energy, for example, or aviation have been using human factors research and human factor specialists for years already. It starts to become more and more popular in medicine. And I think in the case of AI, it's especially important as well. And of course, if you look at the recommendation and even the regulatory requirements to develop new devices, you need to do some form of human factor evaluation before you can bring a product to market. But at the moment, the main requirement is to make it preclinically. And of course, you have to test the usability of a new device preclinically. But we make the argument that as you start using this device in real condition, you might come across problems that you did not foresee before, or you might come across features of the device or aspects of its use that were not anticipated. So we also need to test that again at small scales, but in live, real clinical setting. By the way, when I say live clinical setting, I mean settings where the decision made by a human clinician, supported by the AI system, has an actual impact on patients. Because it's easy to say, oh yes, I will trust or I will follow the decision, the recommendation of the AI system. If that has no implication, it's another question when it has actually an implication on patient care. And I'll come back to this point at the end of the presentation with the very nice examples that was published recently. And the last important part of those early stage studies is to prepare for the larger scale trial. That's one of the main arguments made in the ideal framework, is that you need to define a few parameters of your larger trial during early stage, some call it pilot studies, in order to have robust, well-conducted and successful trials afterwards. For the sake of time, I'll skip this slide, but that was two more examples of why guidance at this stage were needed. So that's how we decided to develop Decide AI, this new reporting guidelines for the early stage live clinical evaluation. How do we do that? How do we develop guidelines that will then hopefully be recognized and followed? Well, we need to gather opinions from recognized experts in the field and to bring them to a consensus. For that, there is quite well known and broadly used methodology, which is called a Delphi process. Was originally developed by the US military with a very interesting question of how much bomb power do you think the Soviet would need to bomb specific targets in the country, and which those targets would be. That was the original case study, but then it was applied to a lot more applications. So that's the graphical representation of the Decide AI Delphi that we conducted. So we started with an initial list of reporting items, basically what researchers should report in their publication when they describe the evaluation of their AI system. And we moved through 2 rounds of Delphi to an updated list, which was then presented to a consensus group. Consensus group was the Decide AI expert group. It's about 150 experts. I'll show you a more detailed composition of this group just after. And the Decide AI consensus group, the final consensus group, was a group of 16 experts. And we had the chance to have Dr. Matter as part of those 16 experts. We then piloted the guideline with another group of experts in the field that were not part of the consensus group to see if the recommendation made sense and if the explanation given were intelligible. That's a brief overview of the stakeholders group affiliation of our experts. We can note a very strong representation of clinicians and engineers, but we made sure that to have also expertise and feedbacks from different groups and expertise interested in the development of AI systems. We also made sure that this overwhelming representation of clinicians and engineers did not, how shall I put it, overflow the rest of the conversation. So we wanted to make sure that the underrepresented group were also considered equally. And that's why for all of the scorings that we've done, I won't go into the details here, we broke that down into the different stakeholder groups to see if there were any major dissents between them. The outcome is a 28 items recommendation checklist that are grouped into generic reporting items and AI-specific reporting items. The generic reporting items are the one that could apply to every studies, but that you need to have as a backbone for the reporting checklist. And the other ones are the more AI-specific recommendation that we were making. I mean, if you want to read in details all those items, I recommend the recent publication we had in jointly in the BMJ and Nature Medicine. I'll happily give you the link at the end of the presentation. But if I have to summarize, what are the key novelty, the key component we have in these guidelines? And again, I take the 4 important reasons why early evaluation is crucial in our opinion. So for the performance aspect, we made recommendation to report on the intended use of the algorithm. It's important, as I said, describe both the patient population, but also the user population, because as explained previously with surgery, this has an influence on the performance you will see of your model depending on who use it and what population you apply it on. The implementation setting should be also described quite transparently because that could also have an influence. One of the key novelty of this AI is to understand that modification of an AI system in early stage of evaluation is both acceptable and also beneficial for the development process. A lot of the, if you look at how industry develop products, rapid iterative evaluation design cycles are often used in early stages to improve and tailor your product to the future users and the future audience. And I think as long as it's part of the protocol, it's part of the way you want to evaluate a system, and as long as it's reported accordingly and adequately, modifications are acceptable in early stages. For the safety, the recommendation, we recommend to have a detailed error analysis at those early stages, because that can not only allow researchers to understand where those errors come from, but also to correct them and avoid repeating them in subsequent trials. As I said, and I'm looking at my watch at the moment that we can finish the talk in time. Human factors, we make recommendation about usability, learning curve, quite important. The way the AI system output is represented, and we encourage researchers to report on the human computer agreement. All of those items were included in the guidelines because we and the experts involved think that they can influence and hence also bias the overall results presented in the study, depending on how they were set up and followed. I just wanted to briefly touch upon some important topics that were excluded during the process, because I think they're still important to discuss in the context of AI evaluation in clinical settings. The first one is the standalone AI performance. The final recommendation was that in early clinical stages, it's not important anymore to see the standalone performance, but it's the assisted human performance which should be reported. There was some disagreement among the experts. I think that's a topic that needs to be further researched and debated. The interesting aspect, if you have the standalone AI performance and the assisted human performance, then you can also start quantifying what's the impact of the human brain in the decision-making process. And I think that's quite valuable for further research. Interpretability was not included as well. That was a quite controversial one because you could see two groups, people that are strong advocates of having interpretability as a basic feature of any AI system, any successful AI system, and others say, well, interpretability is after all not that important. I mean, if you have a very accurate, a very efficient system, it doesn't really matter if we understand why and what the system makes. I'm not taking any sides here. I'm just saying it's an ongoing debate, and I think that might come back later on in the future iteration of the guidelines. The last point, and for me the most interesting one, is the trust curve. I think this item was not included at this time because we lack ways of measuring trust in a reliable way, and hence you cannot force authors to report on trust and evolution of trust if we don't have broadly accepted way to measure trust. But I'll just give you an example why I think it's so important to have it long-term in AI evaluation reports. And that's one of my favorite paper was from McIntosh and colleagues, recently published in Nature Medicine as well. In the context of, they developed a machine learning system to make recommendations for radiotherapy planning in the context of prostate cancer. And what you can see here is that, and basically they did two phases of their trial. The first one was a purely simulated phase, and the second one was a real deployment phase. In the purely simulated phase, they were presenting one radiotherapy plan made by a human radiologist and one made by their machine learning system. And they were asking a panel of physicians, which plan do you think is superior? And then which one would you apply to your patient? And you can see that the quality of the radiotherapy plans proposed by the AI system was equivalent in both the simulation and the deployment phase. Yet, when it came to the deployment phase, you could see a marked drop in the number of those plans that was on the machine learning plan that was selected by the human clinician and then applied on their patient. So they consider the machine learning radiotherapy plan equally good as the human-made machine learning radiotherapy plan, yet they would not apply it to their patient. The question is why, and that's for me a question of trust. That's a trust gap in the actual live application of AI in clinical setting. And I think that's a very interesting further field of research if we want to understand what happened and how to correct that and and help bring trust from clinician and patients and hospital manager in AI system and allow them to deploy their full potential to improve patient care. Thank you very much for your attention. I'm very happy to take any question.
Yeah, thank you, Baptiste, and thank you for thoroughly explaining the background and how the Decide AI came into being. And I know we are short of time, but would love to take at least a couple of questions that are already there in the chat. The one is from Paul Manberg, and his question is to Baptiste. He says, I'd be interested to hear how you plan to track adoption of this guideline, as well as how quickly you expect these to be reported on, say, a threshold of 50% or more of the study publications. So any thoughts on that?
Oh, thanks for the question. You identify correctly, that's the major challenge when developing reporting guidelines, it's acceptance. Even guidelines like Consort that is the mother of all reporting guidelines, a very robust, a very well accepted one, is still, I think it's still far from 50% of study using it to report it. It will depend as well on how many journals will endorse those guidelines and have it as a requirement for publication. That's the work we're starting to do now, is to contact journals to convince them to accept and endorse Decide AI as a requirement to publish with them. that will take a long time, and I think I cannot advance any number on that. I'm sorry.
So I'll just add to that, that while at the beginning I showed you our year-end reviews and we showed the quantitative assessment, there is a lot of work that is going on qualitative assessment of maturity of these publications. And we are working with some groups on the same. And we would like to see how these guidelines are getting adopted and be able to report on that in the future too. So stand by, that work should be coming soon. Going back to Dr. Stevens and Shubhayu, I think some really interesting work related to the GCS motor component assessment. I see like a lot of other sensors being added. Are you also trying to look at other GCS components? So there are a couple of other GCS components, both on the sensory side and the verbal side, to get a comprehensive understanding of a patient's mental status. Although some of the work that Ashish is here on the line and I had done and we had reported in critical care showed that the motor component was definitely more predictive of patient outcomes compared to sensory and verbal. So what are your thoughts on how do we pivot from clinical assessments to sensor-based data? It's like flying these planes manually to trying to fly a jet just like Tom Cruise flew.
Thank you for that question. So we are, I'm pursuing right now a new study, which is entirely focusing on the recordings of patients in the ICU using a smartphone. So essentially we're recording the facial expression, as well as the voice. And we're submitting those vocal and facial time series to different deep learning algorithms to see whether we can decode information on underlying physiological states. So yes, and so that sort of ties in with what you were mentioning about the other components of the GCS. So those are that we didn't discuss today, which are the verbal and the eye scores. But it sort of takes it to a different level because we're interested in actually decoding eye movements and facial expression as potential biomarkers of underlying physiology. We hypothesize that people with latent, imminent and impending physiologic decline might actually express that in their facial expression. So that work is ongoing. We just received a grant to support it. We have IRB approval, and we're beginning to enroll our first patients.
That's great. Congratulations. There was a question for Shubhayu, so which was directed in a chat to me, which was related to what is considered high frequency. So how would you determine what frequency is okay for a particular sensor when we're trying to make these assessments? Or how do you determine those thresholds?
Absolutely. That is something of itself that is an important variable in testing these sensors. For us, the accelerometers we use, there's a payoff between frequency and battery life duration. And so it is possible to achieve higher frequency. I think anything above 10 hertz is recommended for activity classification with accelerometers. And so we use that as sort of the baseline to extend duration of measurement. However, it depends on the modality as well. I think it makes more sense to start with lower frequencies, even if you have the data for higher frequencies, to identify a baseline at which a frequency of a certain modality is able to capture that signal for a multitude of reasons, not just battery life or duration of sensing, but also it directly affects the cost of the device and integrability. So those are the I think the primary considerations for us in setting the sampling frequency.
But I hope you would agree, we clearly need to pivot away from the very or extremely low frequency clinical assessments that are made in the hospital, right?
Yes, absolutely. There's a bit of circular logic that has to take place first, because in order to engage or improve the integration of these sensing modalities, we have to compare them against the clinical assessments to show that, yes, they're able to capture these changes and whatnot. But ultimately, the goal of these is to enhance those assessments, to move beyond those. So, the hope is that one day something like a sensing modalities itself uses the outcome and not just the variable to predict a clinical assessment. But this is the first step in showing that there is that relationship. But we agree with you, yes.
And Baptiste, we are ready to apply Decide AI as Shubhayu and Dr. Stevens continue to work on these AI algorithms, right? And evaluate how good the work is and if it's ready for early stage clinical application. That's right. That's correct, right?
Sorry, if I didn't get the question. If I can evaluate?
Yeah, well, I think Decide AI would be perfect guidelines to apply and evaluate yes. the algorithm that are being created by Dr. Stevens and Shubhayu Bhattacharyay and are going to be coming to clinical settings.
That's correct. And one of the projects we're trying to develop at the moment with Professor McCulloch is to have some form of advisory service for researchers that wants to apply Decide AI, that wants some guidance at the stage of moving from preclinical to clinical studies, as they're doing for IDEAL, and we're considering widening this service to Decide AI. So, but anyway, I'm always happy to talk about Decide AI to give any advice if I can. So yeah, please be in touch.
That's great. So on that, we will end this session. Thank you very much, Dr. Stevens, Shubhayu, and Baptiste for the awesome presentations today. Please stay connected, join BrainX community either through the webpage or through the LinkedIn group. And we look forward to future sessions. And to read the publications that were presented, again, they're listed on our webpage, and you can feel free to contact the authors there. So thank you, everyone, and have a great day.
Thank you.
Thanks a lot. Bye-bye.
Thank you. Bye-bye.